We took some community questions over the last week, as well as they've been just coming in online off the chat and off of the questions. So there's a ton, and I will try to prioritize, but we're going based off of the most number asked. So we'll try to give everybody a shot. There's just so many questions. So the first question I have is for Dr. Wilson and Dr. Dindot. So I think it requires a geneticist and someone who has experience with gene therapy. But if anyone feels that they can answer this question, feel free to jump in. So can you explain if you have any concerns about how you are testing or considering overexpression of paternally expressed genes for UPD? And the reason that that question went to both you, Scott, and Jessica, you probably can jump in on this too, and Dr. Wilson, was in the monkeys that received UBE3A as a human gene replacement that obviously are not missing maternal UBE3A. Were there any symptoms that would support overexpression? And if we are not concerned about overexpression in an animal model, how does that make you feel about a paternal activation approach for overexpression in UPD if you activate both copies of the gene? So Scott, maybe you can answer the both copies of the gene, and Jim, you can answer what the monkeys seemed to experience.
So I'll think about carefully what I'm going to say here. So if you, in theory, if you were to completely reactivate both alleles in UPD, based off the results we have to date, you might expect something less than a twofold increase in protein. And there's some evidence that there's regulation of UB3A when it gets to be too much, but you know from DUP15Q patients that you can have excess amounts of UB3A, and that's thought to be detrimental. To my knowledge, there's no evidence that something less than twofold would be detrimental. I'm trying to think of model systems where that's been done in. And that's in a situation where you're getting complete reactivation of both alleles. In reality, what's likely to happen is it would be a certain percentage of that. I think they may actually be a better candidate because of the amount that you need to knock things down and then get full reactivation. But that would be my answer.
That's a great answer. And I'll add to that, there was a great presentation at the Angelman Syndrome Foundation conference by Ipe Elgrisma looking in his lab. They looked at both maternal and paternal activation, and they showed that there was no phenotypic concern in the mouse model, or in the cells, where they were looking at phenotype in the cells. So that further supported that there was less concern about what could happen, and that was with ASOs, but for UPD genotype, which was very promising as well. Dr. Wilson, do you have any comments on what you experienced?
Sure. I think in any of these approaches where we're using vectors or editing, what we're likely to see is kind of a patchwork of expression or reactivation, as opposed to every cell expressing an increased amount of UB3A. And I'm not exactly sure how to extrapolate from that to a situation where genetically there's overexpression in all cells. But in terms of the monkeys, what I'm pretty confident we have are a subset of cells, a minor subset, that probably overexpress UB3A, but that many of them express very low levels. We don't have a situation in monkeys where we have a delivery vehicle where we can really achieve super physiologic level in all cells. But anyway, in the context of what I just described, we do look at, as best as we can, any neurologic or behavioral abnormalities in the monkeys, and also study the animals for specific neuropathic effects. And in the animals, we're completely fine.
That's great to know. And I think along those lines, Dr. Duis, yes, UPD is very excited. Lauren, I got you. These are all from Lauren. But that's okay. This is very important, your whole community of UPD, which we don't want to leave out of anything. So we want to make sure that's heard. Dr. Duis, this may be for you, in your clinical experience. Have you ever met a patient with deletion of many or some of the other genes outside of UBE3A that have UBE3A, that have symptoms related to the fact that they're missing the other GABA genes or some of those other 10 genes outside of UBE3A?
So I hope I answer this because I'm not sure I fully understand the question, but, or got the whole question. So we certainly see kids who have mutations in some of the GABA receptor genes, and they do have epilepsy. So we see people who have isolated mutations in the genes within the deleted region of chromosome 15. That's the most common deleted region in both the class 1 and class 2 deletions. In terms of the genotype-phenotype correlation, so that means when we're looking at individuals who have a class 1 deletion or a class 2 deletion, there are some things that I don't think they're well distinguished, and there's some discrepancies in the literature, but I would say there are some slight differences that I think we see, having seen quite a few Angelman patients. I don't know if that answers the question.
Yeah, well, it wasn't about UBE3A. It's are there patients
others
that are missing all the other genes and not UBE3A? So we can try to understand
deletion.
the phenotype once UBE3A is replaced, what we should expect in deletion patients when they have UBE3A replaced, which you pretty much did just answer, I think.
Yeah,
in the first.
I mean, from a mutation standpoint, yes. So within that region, there are many genes, and we have kids, particularly with epilepsy, that we follow who have just those mutated genes. I've never seen anybody with a deletion in the region who had presented to us with a deletion that didn't include UBE3A, though. So nothing perfect to really tell you the answer to that question.
It's close, though. We'll take just epilepsy. I'm all right with that. Okay, Scott. Scott thinks he can also contribute here.
But are there gain of function or loss of function in the GABA? They're mostly gain of function.
Mostly, I think actually the ones I've seen have been loss of function, because we have some autosomal recessive patients that we follow.
So that's an important distinction, the gain of function or loss of function.
Because they both can cause epilepsy, potentially. Yeah, that's a good point. Okay, so that's promising, because we have anti-epileptics. We're okay. Okay, next question is for Dr. Nolta. The question was, for those individuals and families looking to have more children, do you recommend that they consider banking their cord blood or tissues, and if that could be of benefit to potential stem cell therapies in the future?
That's a great question. For the foreseeable future, we're going to be going with the bone marrow stem cells. So it might be useful for other things, but I think for this clinical trial, it's the bone marrow.
So the answer is you can save your $5,000 and put that toward donations to invest in research to cure Angelman syndrome.
Or you can donate it to a public bank where others who need it, who might have leukemia. That's mostly where the cord blood's used.
Yeah. I still have mine deep frozen. Okay, another question here is for Dr. Yong-Hui Jiang. What are you seeing in the full, well, I can answer this actually because we don't have it characterized yet. They wanted to understand the characterization of the mouse model, which is only just being made, so we can't answer that, I'm sorry. Dr. Butler, what is the difference between CRISPR, shRNA, and mRNA for paternal activation?
So compared to what we're trying to do with the shRNA, typically, the mRNAs, there's less chance for side effects, things like that. With shRNA, it can be more efficacious, but there's more chance for a side effect. In terms of CRISPR-Cas9 or CRISPR, any type of gene editing, what you have to take into account is that there's also off-target possibilities with the guide RNA as well. So in terms of efficacy compared to siRNA, I don't really know head-to-head. My instinct is they'd probably be similar. I think does that answer it sufficiently?
Yeah, I think mechanistically it's similar in terms of what we're trying to accomplish. But I think
Mechanistically, so at least with ASOs, they have all sorts of modifications where they can go directly to the nucleus, and that's really ideal. In terms of shRNA and mRNA, historically, they would say that siRNAs and shRNAs were more cytoplasmic, but there is more recent evidence that it does localize both to nucleus and the cytoplasm. So, and also in terms of how it degrades the RNA, that is different, but it's mechanistically not that. It will work. I mean, it will degrade the RNA as you want it to, just the actual mechanisms are slightly different.
Well,
Dave, you want to jump in as well?
I think there's one important difference between antisense oligonucleotides and siRNA is that one can be vectorized and expressed from something like an AAV vector, whereas ASOs, by their nature, are generally not able to be expressed, so they have to be re-administered periodically.
Correct. But what the audience needs to know is that they all activate the paternal copy of the gene, and some may be one and done that are carried in by an AAV, and some are going to need to be repeat dosed like an ASO, where it is really just a more short-lived type of therapy. I'm translating. This is for Dr. Wilson. You are working on every approach for Angelman syndrome gene therapy, including maternal replacement, paternal activation with CRISPR, and with mRNA. Which approach do you feel is best for Angelman syndrome human patients? I'm sorry, but I have to ask that question.
Sure. You know, you can hear me okay?
Yes.
Can you hear me? Great. The approach that I'm more comfortable with now is a gene replacement approach. And in that, I believe that it's likely to be that we will know more about the safety of that approach than anything else, because we have, 40 years of gene therapy experience, and we're leveraging many other trials and many other laboratories. There is a concern, though, about ultimately the durability of that effect, even though it's one and done. Does one and done mean 1 year, 5 years, 10 years, 20 years, 30 years? But so far, when we've studied this in monkeys and other programs, there's no decrease by, within 5 years. But the other limitation is you have far less control over what the level of expression is going to be from a vector. The editing approach may ultimately be the cleanest, but we do not have a delivery system for us to really bring editing into the clinic. So I would say at this point, what we would feel more comfortable with would probably be a gene therapy approach in which the UB3A was capable of cross-correction, secretion, and reuptake, because we have brought are bringing into the clinic 5 programs of AAV through an ICM approach, expressing an enzyme for which there's cross-correction. So if we can leverage that experience, that would be awesome. Working with our colleagues at Amicus, we have to convince ourselves that this modified version, UP3A, is secreted and taken up by appropriate cells and functional. But others are working on this, and others have promising data. I would suspect that may be the one that we would bring forward first.
Okay, thank you for that. This one is for Dr. Dindot. A couple of funny questions, but they're wondering when you are going to get the Angelman syndrome pig to UC Davis for their infrastructure so that they have that animal model. Number one, which I'd love to hear the answer to that, because it looks like Davis can handle pigs based off of Dr. Nolta's picture of UC Davis. Is the first one. I'm going to have both, and then you can answer. And then the second one was also based on that, which I do know the answer. if you announced today that you are working with some companies with your pigs, which is really exciting, is there going to be an opportunity for you to share tissues and CSF and blood with other partners, especially the ABOM for the Biomarker and Outcome Measure Consortium?
Yes. So with regards to the first question, any facility that is set up to house large animals, so UC Davis has vet school, I mean, it'd be no different than, it's a little bit different, but it'd be really no different than transferring mice. And then two, yes, the goal here was to get the model up and running and do some characterization of it and get to that point where we could do things. And we're open for business, not business, but collaborations with everybody.
Wonderful. So contact Dr. Scott Dindot if you want CSF, tissue, or blood. Sounds great. Okay. This is a question for Dr. Dr. Nolta. How supportive in your obvious tremendous experience with the FDA is hematopoietic stem cell gene therapy for patients with rare diseases by the FDA? Is this an approach that they are supporting?
Yeah, they're very supportive, and they work with us all through the process. So we approached them early, as you heard from Dr. Anderson and Dr. Abedi. We approached them early with the pre-IND package, and they're very helpful and very supportive. I don't know if Joe or Mehrdad want to add anything to that.
Yeah, I think you all have the same.
Yeah, we meet with them frequently. And
yeah, that's awesome. It's more new to us. We've been hearing about AAV for 5 to 7 years, and so for us, it's exciting but very new. If you were, okay, this is hard, but I'm going to ask it. If you were standing in front of a bunch of potential donors, what would you like them to be most inspired by to donate to your research for Angelman syndrome? So I'm going to go down the list. So what in your lab are you most excited about is ultimately the question. So I'm going to start with Dr. Segal. What are you most excited about that you would be interested in getting more support for?
So we're very interested in the opportunities for molecular therapies because ultimately these genetic diseases are caused by problems in molecules, and our molecular therapy gets at those kinds of issues in a way that drugs don't. And so I'm excited about that possibility. There's been a number of different methods that have been talked about over the last few days, and like Dr. Wilson says, we don't want to think about what's the best shot on goal. We want to have multiple shots on goal, and we're going to see where these land. I'm personally very excited about the work in our lab with Cas13 because we just got a $1.3 million grant to work on that, and it was actually ranked the highest in the state, so we're excited about that.
That's awesome. Dr. Fink, do you have some, why don't we pass it down? Do you have? I'll give it to you, Scott, last.
I think I would echo what Dr. Segal just said, and as well as Dr. Wilson, that being able to work under the funded infrastructure grant where we have the resources, we have amazingly skilled young scientists working in the lab, that we now have the ability to start comparing a lot of these different approaches head-to-head, be it AAV, cell-based, CRISPR-based, that we can start using those and using the same comparisons in mice side-by-side to really understand what's going to be that most appropriate shot on goal. And then, of course, the amazing resources that FAST has been able to pull together for large animal studies, pigs, the UPenn Orphan Disease Center and their gene therapy program to be able to really leverage the expertise regulatory to move these things forward. So I think the thing that I'm most excited about is the very comprehensive research that we can start to look at these things in an unbiased fashion and pick the ones that we think have the most promise to move forward and make the biggest impact.
Well, I actually have two. One of them is...
this is the time to make your plea.
Well, we work really hard to help the day-to-day quality of life for kids with Angelman with what we have available currently clinically, and work really hard on a couple of things, gait, access to communication in AAC, and sleep. And so I have a phenomenal team, and I want more people to be able to access that type of specialty care because my perception is, which could be wrong, that we do make a difference in the day-to-day lives of families, and so I would want to continue to see that expand with where we are with the emergency line that has already been so generously supported by FAST. My second one is that I work a lot on outcome measures and informed by our clinical work, and so I'd love to see some more funneling of funding towards really working for an objective outcome measure that really does span, even if it's one domain, does correlate to other improved domains in Angelman syndrome.
Dr. Butler?
Yeah, so for the gene therapy field, I would say I would most like funding for examining novel viral vectors that are potentially better than AAV9. So a lot of companies, a lot of research labs invest a lot of time and money in developing them. My lab doesn't currently have that aim, but among other things, I have a strong collaboration with Dr. Stephen Gray, who also makes new viruses, and some of them have potentially shown to penetrate the brain better and things like that. I've spoken with people at Voyager Therapeutics as well, and they recently described a whole line of new viral vectors that could potentially be orders of magnitude better than AAV9. And so I would like to see how they work in a disease model. So that's what I would like.
Dr. Keung?
So I think one of the things that's struck me a lot about Angelman syndrome when I started learning about it several years ago was a duality in the fact that it's a monogenic disorder, but it also isn't at the same time. It's very diverse, it's very complex. There are multiple genotypes, but there's also a lot of complexity in terms of timing, genetic backgrounds. We haven't even thought about too much about sex and gender and racial and ethnic backgrounds. So and complexity is something that I think a lot about as an engineer and as a synthetic biologist, which is kind of one of the ways I'd kind of think about our work. And one of the things that engineering synthetic biology does is try to think about really efficient ways to access and capture complexity. And so some of what I talked about today is trying to get at that, but I think what I'm really excited about is that there are there's modern molecular biology tools and analytical methods that are able to access an incredible amount of diversity in cell type and in functional manipulations, and to read out those changes in really creative and high throughput ways as well. And so I think that that's kind of where I'm thinking that our lab can try to provide a unique contribution to really think about how to efficiently access that really diverse and complex space.
I appreciate that answer, because we fund everyone here. Well, we don't fund Ryan actually, but we fund everyone here a fair amount, and it's really exciting for y'all to think about what are your next steps, right? So we fund you and you make great progress, but I like to hear about the next steps 2 or 3 years before you think you needed to think about the next steps, because that's where we have to be. We always have to be 2 or 3 years ahead of where we think we need to be. So I love that answer. Dr. Yong-Hui Jiang, would you like to tell us what you're most excited about and what your next steps would be? He doesn't want to tell us. Dr. Jiang? Just kidding. Maybe Dr. Wilson wants to tell us.
Sure. I'll get his portion of the funding, I guess. So about what your next steps would be?
Sure. I have two sort of I'm most excited. Can you hear me okay?
Yeah, we hear you.
Yeah, can you hear me?
Just kidding.
Can you hear me?
Yeah, I think he has a delay. We hear you. Just listen on Zoom. Don't listen on the platform.
Alice, okay. We hear you. You hear me? Oh, okay.
I love him so much.
You all can hear me? I just kind of got a little bit.
Can you hear me now? We hear you.
Oh, yeah. Oh, okay. So, one quite interesting to explore so the non-viral delivery system, which I don't know what it is, but we are exploring that direction. The second is I'm excited about all this reactivation, antisense, and shRNA. But I kind of feel like we're still missing something here. Which is we know that UBCA imprinting is cell type specific, but we don't really check them very carefully when you reactivate them. And this randomly sort of oligo screening, we are checking expression reactivation superficially right now. Like which type of cells really you reactivating? Are you reactivating right time, right location? So I really feel like I want to explore how the antisense truly regulate this tissue-specific imprinted on exclusively maternal expression pattern in the brain, particularly in the human, would be ideally the mice to jump to the human, probably there is a gap there, but the human with spring organelle probably give us some kind of the possibility, understanding better, starting with mice, but eventually we need an understanding how the antisense oligo, or antisense per se, in general, to regulate the brain or cell type of specific imprinting pattern.
Thank you for that. And then we'll have Dr. Wilson and Dr. Dindot go. So Dr. Wilson?
Great, thank you. Really two areas. We'd like to be able to put Angelman syndrome at the head of the line for our in vivo CNS editing our global neuropathic program, in that we'll have funding to develop the non-viral delivery strategies, but it's useful to have a disease model that you can demonstrate proof of concept. And since we have some data with AAV, that would be helpful. I think once we generate those data, we'd be able to get a lot of interest for additional funding. And then as well, we have preliminary data on some activity of a microRNA, but we have a ways to go. And additional support to move that to the point where we have proof of concept and begin to think about translation to the clinic. So those are the two areas that I would be most interested to further working with FAST on.
Scott?
Well, all the good ideas are taken. I could use
cow model of
I could use a new barn. A new barn. And you can get your name on it and everything.
You could just send the pics to Davis. They have barns. Just kidding. Love you. How about a cow?
No.
I love cows. Okay. See, Scott's heart is all in the right place, but you're all doing great work, so I really appreciate all of your answers. We have a few more minutes. So one question that we had was to Dr. Wilson, which was to understand your animal model work, your mouse work with your human UB3A replacement. And the question was how that compared to the work that was recently published from Matt Judson out of Philpott's lab, looking at the two isoforms and how they got more neurons transfected ultimately. And it's a PHPB question. So can you just basically explain the difference between AAV9 in transfection and PHPB in the fact that it's not translatable outside of the rodent?
Sure. Well, in mice, because the brains are so small, that you can achieve pretty efficient global transduction when you inject the vector into the cerebral spinal fluid, which you do not see in monkeys. So we did have good transduction with an injection of an AAV9 type vector into the CSF. But the other approach, which actually may be more efficient, is injecting the vector into the blood with a capsid that penetrates the blood-brain barrier and gets broad distribution into the brain. And a number of years ago, there was a variant of AV9 developed by a group at Caltech called HPV that achieved this with remarkable success in mice that was not translated to monkeys. However, Voyager, who actually had in-licensed that technology and then learned that it wasn't really useful in anything other than mice, has gone back to the drawing board and has claimed that they have a PHPV-like capsid, and we're all waiting to sort of see more about it and get access to it so we can test that. So that's the current kind of short debrief on my view of capsids, Allison.
Great, thank you. And that was the difference between the transection for that person who asked that question. They were different capsids, but one's not translatable. So most of these questions have now all been answered in all of your talks as well. One comment was just a comment, but I think we should say it, was a thank you to Dr. Yong-Hui Jiang, who introduced us to the Chinese Angelman Syndrome community, because there's a comment from a Chinese family online that is thanking FAST for speaking at their Chinese symposium, because they now have over 1,000 patients in China that have been identified with Angelman syndrome. And if it weren't for you and our introduction and their inspiration from our talks at their Congress, they would not have so many parents coming to them in their organization. So I just wanted to make that comment, that you bringing the Angelman syndrome hope around the world has made a big difference in the lives of many, many families. Okay, so I want to see, I have a few more, but I want to see if there's anyone in the audience who has been inspired. Since you are all here and you made the trip, you deserve to ask your questions. Any more inspiration in the audience?
First of all, thank you so much for all your work you do. And my question would be, it would be fair to say that most kind of methods that we discussed today are focusing on the deletion cases. And I'm wondering if you think there would be an approach out there that is not efficient for deletion cases, but would be efficient for other types, like UPD. Obviously, you can tell that I have a daughter with UPD, so thank you.
So I think we kind of answered that as the first question earlier, so I can just summarize that if that's okay. So essentially, I think what people were saying was that all therapeutics may be a great benefit for UPD. So it's not that one is focused necessarily just on deletion, it was just the easiest place to start. But the bigger concern was gene replacement can be great for UPD. Any type of replacement of UB3A could be great for UPD. What is the issue if we activate both copies with a paternal activation? And the consensus in the answers were that there may not be much of a concern, but it's a theoretical concern. And the more data that's coming out in animal models and cell lines is supporting that it's unlikely that even if they got to 200% in these neurons, which would be the most you would get in every neuron, which is very unlikely that would ever happen, it may be in some neurons you get some activation, but you're likely not going to get all of it in every neuron. There still may not be a problem with that, and it may actually benefit those patients more, because if we only get 50% in one neuron for mutation or deletion kids, a UPD would double that. And so the answer I heard from the panel was that it's a really promising data coming out that maybe we shouldn't be as concerned about UPD as the theoretical concerns of overexpression. Does that help summarize the answer? So I think any approach could potentially benefit UPD.
I have a two-part question for Albert. I'm wondering how you envision using the organoids that you're developing in drug development. And the second part is, you touched on it a little bit in your answer about diversity. The question is about the genomic background. So do you envision going from the one genomic background that you're working on to a wide variety of genomic backgrounds? Because for a drug to be successful ultimately, it needs to work in a wide variety of genomic backgrounds.
Yeah, these are great questions. So the first question in terms of drug development, right? One thing I guess I didn't mention too much, but is applicable to the cell systems that we're generating and that Dr. Jiang is generating, is potentially developing the therapies in a human context right from the start. So for example, rather than developing some of the therapies in mice first and then porting them over and redesigning the sequences that you're targeting for human, you could potentially just start that process right from the beginning. And that's somewhat important because oftentimes when you port things from one species to the other, the sequences are completely different that you might want to target, the sequences that are effective, and so you'll need to do screening, and that can take some time. So from a screening perspective, as well as just the development of the therapeutics in the beginning, that could be useful. And then organoids in general, and further kind of drug screening and development. I think one of the really powerful things about organoids are that they are amenable to kind of high throughput approaches and high throughput sequencing and kind of bar-coded systems. And so you can actually even imagine doing assays where you're mixing multiple different cell types and genotypes into and making organoids that are mosaics, and actually screening in I guess what you could call like a single pot, rather than having many, many, many different wells. You could do a lot of experiments in a single pot that's really efficient, and do screening in that way. And your readouts would also be very efficient using kind of next generation sequencing approaches. So I think yeah, those are kind of to answer your first question, hopefully. And then the second question was about genetic background, right? So we're planning to go after H9 and H1 stem cell lines. So these are two embryonic pluripotent stem cell lines that are allowed under the U.S. federal guidelines to be worked with using federal funding and any funding really. And the reason we chose these are one is male and one is female. So we wanted to first capture that, but I think we would definitely want to expand once we have all the tools developed and all of the sequences and targets selected. We would basically want to repeat on iPSCs from different genetic backgrounds, and probably need to confer with geneticists to see what would capture a diverse sampling of the global population.
I actually have a really good question here for Dr. Dindot. I know he's giving me the evil eye. No, it's a good one. It's a good one. So what are the chances of paternal mutations or SNP that could make a targeted approach to paternal activation problematic for individuals with Angelman syndrome?
So you could actually estimate what that is based off the occurrence of mutations that cause Angelman syndrome. Right? So they would just be coming from a different parent, and it would be extremely rare, right? It'd be like lightning striking twice, I think, for that to happen.
So 10% of 1 in 15,000, essentially.
Yeah, because
mutations are 10%, and then
you would have to have a situation where mutations were a deletion, for example, was inherited on one allele, and a mutation was inherited on the other. It's all the scenarios that are possible would have been likely both being de novo mutations that are very different. I just think the odds of that happening would be extremely rare. It's a great question, but
we have sophisticated parents. That was a good question.
So is the question whether a mutation would render the therapy inactive?
It was basically if we would need to sequence the paternal side of patients because an ASO or a gene therapy or some paternal activation approach that's sequence specific might not work because they have a SNP or they have some other mutation on the paternal side that won't then have a match with the therapeutic.
Yeah, I would agree with Scott. It's going to be much less than 10% of 15,000.
Yeah, I was thinking, well, 1 in 15,000 on one side, 1 in 15,000 on the other side, and then a SNP would be even.
Yeah, but it'd be 1 in 15,000 times.
Times 1 in 15,000, and then
10% of that.
10% of that, which is obviously really, really low. Yeah.
But I think the question was a good one about there's the therapeutic. I mean, if you're talking about an ASO binding to its site, there has to be perfect complementarity. And there's also a chance for there to be a mutation that affects the ability of the sRNA, the microRNA, the shRNA, or the ASO to bind to the antisense transcript. And so the approach that is often used is just to make sure that you're targeting a region that is not polymorphic across humans, so that it's a sequence that you're somewhat confident that will never change, or the odds of it changing are extremely unlikely. I think that.
Because it's not targeting UB3A, it's targeting the antisense. Yeah. You have a comment on that, Kyle?
There's a question over there.
Oh.
We have a question.
This is a safety question, and specifically for Dr. Wilson. It seems like the DRG pathology was a serendipitous discovery, and you are avoiding that by elevating the site of injection to, the somewhere higher in the spinal cord. How certain are you that you're not just relocating the pathology to some other unsuspecting cells in the brain, or is the DRG so specialized that you wouldn't expect something like that to happen in other neuronal cells?
Yeah, it's a good question. We were surprised when we first saw it, and what we subsequently learned, that no matter how you inject the vector into the cerebral spinal fluid, a lumbar ventricle, or ICM, that the one cell type that takes up more vector than any other cell type are the DRG sensory neurons. And the problem is really, which is kind of unusual in gene therapy, is too much vector expressing too much transgene. So in order to achieve more gene transfer outside of the DRGs, you inadvertently over-transduce the DRGs. So we don't really see that problem because we don't have the problem of over-transduction in other parts of the brain. We do have a mitigating strategy that I didn't have a chance to tell you about that we've now licensed to, I think, 5 companies, which is we incorporate a sequence into the vector, that a small sequence that is represented in the RNA that then shreds the RNA only in DRG and not in other cells, so that we can actually ablate any expression in the DRG of your transgene. So if you have a disease where you don't need expression in your DRG, this is a pretty generic fix.
Thank you. So for the sake of time, I'm going to ask each one of you, as we do every year, to speak to our families who have been with us for 8 hours and are still sitting in their seats with their eyes open, most of you, who want to hear one message from each of you for the future for their child. And if you want to say one message to them on how they can get through the next year until we have another robust update like this, but we'll have more coming soon, sooner than a year, this I promise you. What would you say to our families?
Just thank you. You inspire us every day. This is why we do this research. And that's just why we do it. So just thank you. Thank you for all your support, for raising the funds, and just, the pictures and the video of the kids. That's what keeps us going.
Thank you.
And so I echo that. I mean, it's very inspiring to be here. And I think that, we all go back and work a lot harder, and our students and our trainees see that, and that they're inspired by that. But there's a few of us have been around long enough to kind of see this grow over the years, and it's really turned into something amazing. So I think that it's just going to continue to get bigger and better. And I mean, you look at all the work that's being done; it's pretty remarkable what's being accomplished here. And so it's just going to grow, and things are going to get better, and we're going to learn more. And there's just a lot to be hopeful for.
Thank you.
So I would say the same thing, but I'd say it in a different way.
Of course you would.
The ability of FAST to fund science and then go on to make a company that made the very first clinical trial of a molecular therapy for Angelman syndrome has really profoundly changed the course of the history of this disease for the very first time. And that's just the beginning, you know, because all these other things are coming up behind it. So I would say that there's never been a more exciting time in the history of Angelman syndrome as there is now, and it's only getting better.
Thank you, Dr. Segal.
Yeah, I don't know what I can add after those outstanding sentiments, but to continue to be optimistic, to continue to look forward. I think as you've heard today of all these different therapeutic approaches, they're similar but different enough that you know, if a roadblock is met in one issue of delivery or transgene expression or reactivation, there's a whole other menu of things that are moving just as quickly forward. And so this shots on goal idea that we've all echoed a few times is continue to be optimistic. There's many, many wonderful labs that are working at this, at pushing this forward as fast as possible. So,
thank you.
Well, I would echo all the sentiments and say thank you. It's a pleasure to work with this community, and thank you to FAST for accelerating it and for all the hard work you do. I mean, I would also say this is happening, and Angelman syndrome is poised to be one of the first central nervous system neurogenetic disorders to have real personalized therapies, and it's amazing to be along for the ride with the families.
Thank you.
Wow.
I told you, welcome to the community.
Yeah. Well, I would just say, you know, from my professional opinion, that genetics is just straight up cuckoo pants sometimes. So I'm speaking as a, you know, I work on rare diseases, but I'm also a patient with a rare disease, Charcot-Marie tooth. And I just wanted to say that there's, if you've ever thought it, it's not worth divulging it, that it's like karma or something like that. So just continue the fight, and that's all.
Thank you, Dr. Butler.
I think, you know, I think this meeting, one of the reasons I love this meeting is because there's so much inspiring work from the families and from this, you know, scientists, and I'm, you know, pretty junior relative to this crowd. So I, you know, maybe what I would say is that when I look at the people even junior to me that are in the labs, I am very, you know, I'm very optimistic because I think they're way better than I am. And there's a lot of hope because of that, because they're up and coming and they're coming up with all of the new ideas. And so there's a very bright future because I think our young scientists that are up and coming and training right now are so bright and so passionate. So I guess that's what I would say. Yeah.
Dr. Abedi?
I wish I was sitting in the beginning. I'm running out of words here, but I just want to say there's a light at the end of a tunnel, and we're just getting so close now, I think. And there's something about Angelman I think is different, because I review all these data because we want to implement clinical trials. And you know, most of these things which says, you know, maybe we should do it in utero or early. And the data that we're seeing, you know, in even adult mouse, they really reverse the problem. They're just not, you know, staying the course and not getting worse, as we see in other diseases. Even in adult animals, we see that they're reversing the problem. So even for patients who are, you know, older, and the concern for many other diseases was just too late, in Angelman, it may be different. So, and I'm just saying that's a really big hope that we have, that we may have a treatment, even in more established patients, that may reverse that, and I think that's very promising. And we're so close, so close.
Dr. Wilson?
Yeah, thank you. I view where we are now as intersection of three sort of issues. One is a disease for which the genetic basis is complicated, but provides the deployment of unique molecular approaches. The second is, you know, for us that have been in science, I think we're stunned by the development of new technologies. I don't think any of us would have expected that we would have the tools that we now have. And the third is the community, FAST and the patients. and I work with a lot. This is unique, and at the upper 1% or more of foundations that believe in science and believe that science is the answer. And when they talk to us about science, they want to hear about what may be possible, not what is feasible. And you bring those three things together, and I know of no other community that has opportunities for real transformative solutions than those in the Angelman Society. So kudos to the community and leadership of FAST for seeing that, having the vision, and bringing us all together.
That means the world to us. Thank you. Joe?
Well, I'd like to say thank you to everyone always supporting us. It's always great working with everyone, and to also be just looking forward to all this work that everyone's doing, the potential trials that are coming up, and to stay, optimistic and look forward to all the work that everyone's been doing.
Dr. Jiang?
Yeah, so I was a second-year graduate student when the gene UB3 was discovered in Dr. Rodell's lab. I contributed a very small portion. So as you know, I see the patient in the clinic once a week, and then back to the research lab. I always told myself, the motivation of doing research, always see the eye, like many of you, in the clinic. Every time I see your eye, and then it's always motivated me and my lab people. So I really feel after almost 20 years, when I experienced a lot of almost kind of among the panel, I probably experienced the most early exposure for the Angelman history. And but I really see I would say keep the hope, and then the hope is very, very real now. Compared 20 years ago, I wasn't clear when I stepped in doing Angelman research. And it's a hope is very, very reachable because all your efforts, Allison or many other, and you are in the audience for this effort. So, but it's still a challenge. I know it's a lot of challenge ahead of us, but ahead of you, daily life. But the hope is really reachable, as you can see, and you motivate us all the time. And just prepare, ask the hard question for scientists, and we're working harder to working together with you, kind of reach that final goal.
Thank you. And so I'd like to make one comment on behalf of our community to all of you, and then we'll end it and go get drinks. And that is that most people sitting in this room didn't make the choice to work on Angelman syndrome, but every one of you made that choice. You don't live with it. You don't have to do this, but you chose to do it. And we are so grateful to all of you who make a better potential future for our kids, because we have no choice. We have to fight this fight, but you don't. So we thank you for that. Thank you for all you do.